So our next speaker is Dr. Kankshita Swaminathan. She's the research leader in feedstock genomics at the Energy Biosciences Institute, which is a joint institute of University of Illinois and UC Berkeley, and she's at Illinois. Kankshita is probably one of the leading expert in the genomics of a crop that she'll tell you about called Miscanthus. Miscanthus is a giant grass that's one of the premier candidates for biofuel feedstock. But we know very little about it, and it's a little bit of a similar situation to the way Steve described it. A few years ago, we knew almost nothing about the genomics of this plant, and Kankshita and her team have really been driving this forward to really put it at the forefront of biofuel development. Kankshita is a plant biologist. She got her PhD working in Arabidopsis at Dartmouth, and as I said, she's been a leader of this biofuel feedstock genomics program that's been really flourishing at the EBI. She's been heavily involved in using both genetic sequencing and bioinformatics, as we'll hear, but also bringing her knowledge of plant biology to really understand what it is that makes this perennial grass tick, and is learning a lot of interesting things just by studying this grass about perenniality in general and the way in which plants regulate their resources throughout their life cycle. So I'm excited to hear talk today about the genome biology of Miscanthus.
Thanks, Dan. That was really flattering. I'm actually really excited to be here and tell you a little bit about our work with Miscanthus. Actually shown in that title slide is Miscanthus right here next to switchgrass. Miscanthus is actually an introduced plant. It's native to Asia, whereas switchgrass is a native biofuel grass, both of which accumulate a lot of biomass and have great potential. Not hear me? Oh. There we go. Okay, I'm going to begin with thanking the group that's responsible for most of the effort that I'm going to talk about today. It's a group that's split between the Energy Biosciences Institute at Berkeley and Illinois, as Dan pointed out, and the JGI too. So we have a mix of grad students and postdocs from both places. Now, Miscanthus belongs to a tribe of grasses we're all very familiar with. It's called the Andropogoneae tribe of grasses. This includes maize, sorghum, and sugarcane, three large leading biomass and food crops. One thing interesting about this group of grasses is they all seem to be very amenable to breeding, and we've been able to actually make them yield very high amounts of either food or fuel. The other interesting part is that they have a range of adaptations. So sugarcane is a tropical plant, and Brazil has really made a success story out of it for biofuel. Miscanthus is temperate and has a very wide range, so it has more potential in the temperate US. Sorghum is drought tolerant, and given that water is becoming scarcer and scarcer, that's something that's going to really play a role in the future. And maize, as we all know, is a leading crop in the United States and is very versatile. The maize we know is mostly grown for food. However, it can be bred if we want for fuel too. They have a versatility of carbon forms going from the sugars, and sorghum is a nice example there because you have the grain in the grain which has mostly starches. You have the lignocellulosic stems, and in the case of tropical sorghums or sweet sorghum, it can actually put sugar into the stem. So you can actually modulate the kind of sugar that these plants produce. And so they're a nice little group for comparative genomics. I will focus my talk today mostly on Miscanthus. We mostly have seen Miscanthus, although when I started working on this in 2008, I had no idea what I was getting into. I looked in GenBank. There was almost nothing in GenBank for this plant, and I had never seen this grass. But ever since, it's in everybody's lawn. It's in everybody's yard. It's in all botanical gardens. And it's been in the US for a long time. It's just that it's been grown as an ornamental. So if you look around, you will see this grass grown mostly for its leaf shapes, variegation, nice clumping grass, which requires little input. It's perennial. It comes up year after year. And so this is something that I stole from the internet. But these are our, this is a common garden we established in Illinois, shown here and here. And this, again, is the same plot where we're actually comparing the biofuel grasses. This is one of the first plots that were established. Now, the plant that we mostly talk about when we talk about biofuels is actually a sterile triploid hybrid of two species in the genus Miscanthus, Miscanthus sinensis and Miscanthus sacroflorus. It's a diploid Sinensis and a tetraploid sacroflorus, giving you a sterile triploid giganteus, which has hybrid vigor and huge amounts of biomass. Needless to say, although there are a lot of pros because it's an impressive plant, lots of biomass, it appears to be a sustainable solution because it requires little nitrogen inputs because of its perenniality and its rhizome, which I'll talk about more later. But the bottom line is that it tends to recycle its nutrients year after year, putting all its nutrients back into the soil, into its rhizomes at the end of the season. However, this plant was a natural triploid. We don't know who the parents are, and it's clonally propagated. So again, like we heard of the cassava story, if this thing is grown in large scale, we need to bring in the diversity, otherwise we will have a problem. Sterility in a plant that's brought in from Africa or from Asia or anywhere else is always something that we are comfortable with because that means that at least you don't have the pollen and seed spreading and making the plant invasive since we are bringing in something from somewhere else. However, sterility also causes problems because now you have to vegetatively propagate the plant. It has to be, it's mostly by rhizome planting, which increases the cost. There's lack of genetic diversity. However, this actually seems to be very solvable because I know of people like Eric Sachs at the University of Illinois who managed to recreate lines that look like Miscanthus giganteus by crossing Sacroflorus and Sinensis. So it appears to be a very reproducible thing to do. So we can bring in diversity, so that's not going to be a problem. But also, in a perennial, you have to have the farmers adopt it, and that's not going to happen unless there's an industry. Farmers are used to growing annuals, so there's a little bit of a hiccup there. Anyway, we will trudge on because it does seem like a good plant to work on. Now, when we started, we had a few basic goals. The idea was, can we actually use genomics to enable breeding, quick breeding of this plant? And the problems of doing this has been the same for the past 100 years. What we want is traits we see in a huge number of plants, and we want it all in one plant, right? So we want to synthesize a single superplant, which does everything that we want, is resistant to every disease, produces the biomass, and is perfect. Perhaps we can get towards that goal, but we need a few things in place. We need to know where all the genes are in the genome, and what the roles that they play are. So we need a good handle on gene expression. We need to know how they're regulated and how we can manipulate them. And finally, we need to be able to relate the genes or the alleles to the traits that we're interested in. If we have all these things in place, we can actually move towards breeding a plant, breeding our superplant, and using things like genomic selection and transgenic technology and synthetic biology to assemble the perfect plant. Luckily for us, we started in 2008, and that's when next generation sequencing just started ramping up. And this really accelerated a lot of these steps. What we knew at the time that we started was that Miscanthus had 19 chromosomes, was closely related to sorghum, and it had, from flow cytometry, had a genome size of haploid genome size of approximately 2.5 gigs. What we did find out very quickly from these phosmids that were randomly picked and sequenced by Jeremy and Jane at Hudson Alpha was that it's a maze-like genome, which means that you have a few genes in a sea of repeats. Now, this is a bit of a challenge when it comes to assembling something. So you have one gene, and then once you get into the repeats, your assemblies are going to break off. However, you can always get the transcriptome. And so that is what we focused most of our initial generation of resources on. So we used the transcriptome, we used RNAseq data from Illumina to do things like create a genetic map. We picked Miscanthus sinensis cross because that's the diploid, and we had access to these ornamentals. So there was a cross that was made between two different ornamentals, and then we had a mapping population. Now, these plants are self-incompatible, so not everything can cross with everything, and there's a great deal of heterozygosity in these plants. So we have to cross and look in the F1s. The F1s is your mapping population. You can't create an F2. However, it worked brilliantly. So we picked SNPs, variations based on RNA-seq data, using sorghum as a reference. And then we generated what's an Illumina Golden Gate SNP array, which I'm not going to dwell on too long. To make a long story short, we got a complete genetic map, which means that we managed to get all our markers on 19 linkage groups. What was really striking about this was we then put this map onto sorghum, and there was remarkable synteny to sorghum, showing a complete whole genome duplication where every linkage group, there were 2 linkage groups for Miscanthus with the same very highly similar gene order to what was on sorghum, right? Except for 1. So sorghum has 10 chromosomes, Miscanthus has 19. One of that duplication there was a fusion event, so it went down to 19. And we can actually see that in the genetic map very clearly. So this is cool, but also a little bit unnerving because now we had to deal with a whole genome duplication. So really, this is a tetraploid we're looking at with heterozygosity. So we went forward again with the transcriptome profiling. The purpose here was, okay, we had Miscanthus giganteus. We know it's a beautiful plant, lots of biomass. Let's actually get the transcriptome for this. And we proceeded to collect samples through the growing season for multiple different tissues so that we got as complete a transcriptome as possible for a first shot. All of this was done with Illumina data. Illumina, however, changed from being the small little short 36 base pair reads to 76 base pair reads to now 100 and 150 base pair reads and even longer. So our transcriptome had, as we got our samples, we put it through the sequencer, and sequencing technology changed on us. But it still worked out pretty well. When we put our transcriptome back on sorghum, we cover sorghum pretty well. And since we know that it's the whole genome duplication compared to sorghum was what we kept going back to for most of our sanity checks. We assembled this, and it's actually available on Phytosome as a track. The assemblies are available on Phytosome as a track. Now, to get to things that I'm a little bit more interested in, one of the key things about Miscanthus that makes it so successful and also year after year, less input, sustainable, is really the fact that it's perennial. And it owes a lot of this to the rhizome. What is a rhizome? Most of you are probably familiar with ginger, and ginger is a rhizome. What it is an underground stem which grows transversely and becomes a storage organ. Bamboos have rhizomes, and interestingly, ferns have rhizomes, as do some trees. So rhizomes are present throughout the plant kingdom in different species and genuses. And in all cases, they basically provide this little bit of vegetative reproductive success. Obviously, Miscanthus has rhizomes. Remember I told you that Miscanthus giganteus was a cross between a Miscanthus sinensis type and a Miscanthus sacroflorus type, although we don't know exactly who the parents are. Now, Miscanthus sinensis tends to have clumps. It's got a short rhizome. It grows in nice little clumps, which is why we like it for our gardens. Miscanthus sacroflorus we have less of. It's a weedy type, a little bit more scary because what it does is spread well. So that's where all the invasiveness comes from. However, giganteus is a nice in-between. So when these two things were crossed, it got nice, plump rhizomes where it fills a canopy very well, it's nice and dense, but you can still contain it. It doesn't spread as much as sacroflorus. A rhizome is really central to a rhizomatous temperate grass. So let's imagine a plant that has to grow in the temperate season where there's a good prolonged winter. It has two options, to either adapt an annual life cycle or to adapt a perennial life cycle. But the perennial life cycle then has to have a dormancy built in. So you have spring where the rhizomes sprout up, it gives rise to new plants. You have summer where the rhizome doesn't have much of a job, it just sits around while the rest of the canopy fills and grows. And when it comes to fall, the short photoperiods trigger flowering as well as rhizome growth. And now the rhizome actually bulks up, producing more rhizome. This is when the rhizome grows. And then goes dormant, waiting for the winter and just holds off till the next spring. What happens in this process is in autumn, this means that everything from the leaf actually goes down underground. For a biofuel crop, this is perfect, right? Because now what you have left above ground is just carbon stocks. Most of the nourishment has gone back down, which means that you're going to need less nitrogen fertilizer and therefore less pollution going in. It's a plant. It still responds to nitrogen, responds pretty well, but it seems to actually still circulate it. And this is seen really nicely when we look at how the asparagine glutamine free amino acid ratios look like over the growing season. Asparagine is it's a free amino acid that's commonly used in nitrogen remobilization. And we see a little peak in May, which is spring, and then you see a nice big peak in fall, so where it's actually taking it back and dumping it back in the rhizome. And this is just when you look at the rhizome. The leaf and the internodes don't show as much of a profile. So this was interesting. And let's look and see what our RNA expression looked like. I don't expect you to see much here, just that the blues are fall and the oranges and the yellows are spring, things that are abundant and overexpressed in these two seasons. In spring, you basically see response to stimulus, which is not surprising to me. It's responding to the light, the warmth, it's getting rejuvenated, it's all coming up again, and there's a lot of signaling that's happening. In fall, you have certain metabolic processes, mostly nitrogen metabolism. Again, this agrees perfectly with what we've seen before. And in spring, the hormones are raging. Auxin makes perfect sense because auxin's a growth hormone, and it's what it's doing in spring. It's this growth. The other hormone that comes up is jasmonate, and I will come back to this. So again, nitrogen makes perfect sense. It goes with the amino acid profiles that we're seeing, and similar to what we see in the amino acid profile, where we actually see asparagine accumulate, you see asparagine synthetase go through the roof, which is shown in these red bars over here. It goes through the roof in fall, and I had to actually move it into its own own scale because it's tenfold higher than everything else. In spring, you have anthocyanin, which protects these growing buds, and you see a lot of those things come up too. Other things that come up in fall are seed storage proteins, like these lectin domain-containing proteins, which are commonly seen in seed storage. Now, I'm only showing the most predominant asparagine synthetase in this figure. There are multiple. There are 4 in the genome. And there's only one predominant form that still gets its own scale, shown in the gray bars. But there is another one which peaks in spring. And if you remember the little graph that I showed you, there was this little bit of spring expression that you see in of asparagine too, or quantification. Now let's look at the other thing, jasmonate. When I first saw this, I panicked because as an Arabidopsis researcher, usually when you see jasmonate in a transcriptome, you have not collected your samples right. Because what I know jasmonate for is that it's a wound response hormone. So the minute you dig, you're digging these rhizomes up. So I was like, okay, we wounded it, and that's what happened. So we went back and looked again, and it only shows up in spring rhizomes. You can do the same thing to rhizomes the rest of the season, in the middle of the season, in fall. It does not happen. It only happens to spring rhizomes. So it is specific. Okay, and then I looked up papers, and interestingly enough, there are papers where stem modifications like tubers, rhizomes, there are papers where they've actually used jasmonate to induce these things in tissue culture. So it's not such a far stretch. And then finally, this was the final straw, where this paper came out early this year, where they mapped the rhizomatous locus for sorghum. And interestingly enough, when I looked at the genes that were the highest in spring rhizomes and in the jasmonate pathway, they were all, many of them were in this rhizomatous locus. So, I mean, it's one too many different pieces of evidence that suggest that this is something that's true. So it definitely is something that we will look into, and that's where we are right now. Now, in the meantime, we got our hands on a double haploid line, which Kasia had generated when she was a grad student in Poland. Now, this was something that we could now use to look at the genome, because now all variations were because of the two genome duplication copies, and we've gotten rid of the allelic variation that was confounding things on top of everything else. We confirmed that this was a true double haploid by putting it on the same SNP array that we had before. So the parent shows heterozygous SNPs, which are in yellow, whereas the double haploid does not. There's only homozygous. And so we were comfortable with this. And then Therese Mitros has very heroically forged ahead with the help of JGI and their sequencing effort to put together a gene space assembly for this plant. And we're really happy with what we have, because this is mostly Illumina data, mate pairs and different sizes of libraries, but it's all Illumina, it's all short read data. And what we seem to have right now is almost every gene with gene space around it. And now, since it's a maze-like genome, this means that we now probably have regulatory regions, the cis-regulatory regions for just about every gene, which we're really excited about. The other thing is, with this assembly, we can now, for the first time, really tease apart what's happening in the two duplicate copies of the genome. And we looked first at my favorite gene, because it's one of the Jazz genes that are expressed in the rhizome. And only one of the two copies actually shows predominant expression, which is shown right here. And this one is not that much. The other interesting thing is the genes are really highly conserved. Gene order is really highly conserved. However, when you look outside of it, in between genes, there's a serious amount of transposon activity that has changed what the intergenic regions look like. And so now, there are going to be situations where this changes gene expression and regulation, and we have small RNA data that we need to overlay on this genome to get a better idea of what's going on there. So we can start looking at the epigenome too. All right, so we're really happy with the current state of this genome. Now, you would ask, yes, in this case, we have one copy expressed at higher levels than the other. What about in general? So Therese looked at all the scaffold pairs, so the two copies from the two different genomes. She looked at the scaffold pairs, but only for those that had multiple genes on them. And she had about 1,200 pairs to compare. Most of them show that both pairs are expressed at equal levels. So there's a one-to-one. And that's actually nice, because that's what you would expect in most cases for a recent genome duplication. And then you have the ones that are more interesting, where you have one copy being expressed and not the other. So there's some amount of divergence that's happened there. Regulation has changed. And what are those genes? Don't know yet, because this she generated last night. So that brings me to my last slide, which is to just revisit where we started and what our goals were and where we are right now. We wanted to know where the genes are in the genome. We don't know for every single gene, but we do have a complete genetic map and are on the way to actually making a complete one with a lot where we could place pretty much every gene and have more markers than we know what to do with. And then we also have a good handle on gene expression and regulation, and can now begin to ask questions relevant to functional biology, which as a developmental biologist is really what I like doing. And so the other thing that we can do in the meantime, and other groups have actually now started with transgenic technology. There are some reports of transgenics in Miscanthus. However, Miscanthus is in this little panicoid grass group where, transformation is possible in sorghum, it's possible in maize, it's actually possible in sugarcane too, if you want to try. And there's Ceteria. And there's Ceteria viridis, which is a little model plant which grows this high, has a 2-month cycle, and has been proposed as a model organism. And that actually has both the perennial type and the non. And so that's one that is worth trying to transform. So I think overall, we're in a position now to take the next step to actually understanding the Miscanthus genome and what it does best as a perennial crop. And with that, I'd like to thank the group again and all our extended collaborators and take questions.
Thank you, Kankshita. So, Kankshita, this question of perenniality, you sort of showed, and the rhizome, these underground stems that are broadly distributed across the plant kingdom. What's your gut feeling for how universal some of these features are going to be when you think about it?
I do want to look, but I actually think a lot of this might be similar. So my take on jasmonate itself is it's doing the same thing. It's responsible for wounding and pathogenesis response. And in all those cases, what the plant's trying to do is shunt resources away from attack. Right? So it's the same thing. And if you look at or in senescence, where it's moving the resources away from what is senescing to whatever the new sink is. The rhizome is actually something that will senesce in spring compared to the rest of the season. It's senescing and giving and it's providing its nutrients to the shoots. So it might be a program that's actually, and jasmonate's in the entire plant kingdom. So it might be a program that actually, if we look into it, we might find that this part of the program at least was very similar. But it would be nice to look across the plant kingdom, yeah.
Thanks.